Cytosine is our next nucleobase. It is a pyrimidine. So cytosine is the base, and cytidine is the nucleoside. Cytosine is the same cyto prefix that you see in a lot of cytoplasm, cytosol, just meaning cell. So here's cytidine triphosphate. You can see the base here, the sugar here, and the triphosphate here. So this is the RNA nucleotide. Here's the 1' carbon, 2' carbon. So this is the oxygen that would be missing for deoxy cytidine. 3' carbon, 4', 5'. So you can see the 5' carbon connected to the triphosphate. And then here's the nucleobase here. You can see the numbers. 1 is what's attached to the sugar, and 2 will be the oxygen carbonyl group. Then 3 is the nitrogen with a double bond, so it'll have kind of a negative polarization here to attract the positive hydrogen to form a hydrogen bond. And then on the 4th node, you have the amine group. So here's the 4th node with the amine group. So it is a pyrimidine with a single ring of 6 nodes. We call it a heterocyclic aromatic compound. Hetero meaning that it's not just a single type of atom, it's a mix of carbon and nitrogen. So cytosine can go through this deamination process where the amine group is replaced by a a carbonyl group. So you can have cytosine converted to uracil, and we'll talk about uracil later, but cytosine can degrade into uracil. And in kind of the opposite direction, cytosine can have a methyl group added on to this number 5 node here. So that's a way that the DNA can be kind of tagged, just to have some information about, to regulate gene expression, and just to have a way to kind of mark a cytosine. So there's a sequence of cytosine, guanine, just next to each other, called a CPG. And in mammals, 70 to 80% of the cytosines are going to be methylated in that kind of a CPG arrangement. A unique thing about CPG is the complementary bases are CPG as well. So you can methylate one side, duplicate the DNA, and then go and methylate the other side. So it's something you can preserve in the DNA replication process. And lastly, cytosine forms 3 hydrogen bonds with the guanine when it's forming a double-stranded RNA or DNA.